All right, welcome everyone to the 2024 Kansi Distinguished Lecture Series in Statistical Sciences. My name is Sanjina Dang, and I'm an associate professor in the School of Mathematics and Statistics at Carleton University. And I'm also the chair of the nominating committee for the 2024 lectures. This highly prestigious lecture series is delivered by exceptional researchers working in statistics and data science. This annual event features one general lecture and one more specialized lecture. The candidates are nominated annually by a panel of 6 statisticians from universities across Ontario. So the series was launched in 2002, and since then, 22 exceptional research scientists from around the globe have presented their impressive research. And here I'm just highlighting some of our recent speakers. And this year, the nominating committee comprised of Dr. Aisha Ali from University of Guelph, Dr. Jessica Gronsbell from University of Toronto, Dr. David Haziza from University of Ottawa, Dr. Kelly Ramsey from York University, Dr. Michael Wallace from University of Waterloo, and myself. So it is our pleasure to have Dr. Susan Holmes here today to present the work. Dr. Holmes is a professor in statistics department at Stanford University and a highly accomplished researcher. Her research interests include statistics, reproducible research, data science, and statistical applications in microbiology and immunology. She has published over 140 publications, including conference proceedings, has 12 chapters and reviews, and a book titled Modern Statistics for Modern Biology, which is a great resource for aspiring biostatisticians. The many honors and awards received by Dr. Holmes include the Bioconductor Community Award, the IMS Medallion Lecture, the Britain Lecturer, Bremen Lecturer, and the NIH Transformative Research Award. Furthermore, she is a fellow of the Fields Institute and a fellow of the Institute of Mathematical Statistics. She's also very active in the statistics and bioinformatics community. Among her many accomplishments, I'm only going to highlight a few. She is currently an associate editor for the Journal of Open Source Software. In the past, she has also served as associate editors for many journals, including the Journal of Statistical Science, Annals of Applied Statistics, Journal of American Statistical Association, and Journal of Statistical Software. She has also served on the Scientific Committee of Women in Data Science at Stanford, the Science Board of Sydney Mathematical Research Institute, and as a panelist for NIH. As a part of the Distinguished Lecture Series, she will be giving 2 research talks, one titled Hidden Variables Using Statistics to Decode Heterogeneous Microbiome Data today, and the second one tomorrow entitled Statistics and Geometry for Heterogeneous Data, which is, again, scheduled at the same time, 2:30 to 4:30 in the same room. I would also like to invite the audience to join us for a reception immediately following Professor Holmes' lecture. And without any further ado, please welcome Dr. Holmes.
I think he's going to turn mine on, so that's good. Thank you very much, Sanjay, for such a nice introduction, and for the nominating committee for choosing me. I'm not traveling anymore, that's what I said, but I did accept this one, and I'm going to be completely retired starting the year after next, but for the time being, I've been doing mostly work with the Gates Foundation, so I haven't been doing the exact same kind of statistics that I heard in all the talks, but much of what you're doing is very useful. So I'm going to try to explain to you why it's useful to be a statistician when we're in the applied world. Okay. I'm told that in Canada, you can only use Microsoft machines, so I wasn't allowed to use my Mac. Okay, here we go. So this is a story about the work I've been doing over the last 15 years. And I decided that I wanted to understand more about the microbiology, the human microbiome, and immunology. And today I'm going to talk to you mostly about the microbiome data that I got interested in. And I try to think of myself, this is a bridge near where I grew up in the south of France, which is a Roman aqueduct. And there are three subjects that I'm interested in, the three parts of the bridge, and that's statistics, biology, and mathematics. So in what I'm going to talk to you about is we look at the statistics of microbial communities. We have new types of data that we get with next generation sequencing. And there is a lot of information that is lost if you use the standard bioinformatics pipelines. And I'm going to explain to you why thinking more probabilistically has given us much more power and to denoise the data and to think about it. It's useful to think that the data are actually infinite dimensional. In this case, we have many more features than we have samples. And so in some sense, you know, for a statistician, infinity starts when you have P larger than N. And we have some nice hierarchical models which seem to fit pretty well. And we do a lot of in silico generative data simulation in order to do experimental design. And then I'll introduce the problem which motivates what I'm going to talk about tomorrow, which has to do with multi-domain data integration. So this is all joint work with many of my quite patient postdocs and graduate students. Patient, I say, because I don't allow them to publish very much, because every paper, as you'll see, involves a large piece of software, and the software takes a lot of time to develop. And so that's why they're patient. My collaborators are patient because when a biologist asks you a question, they want an answer in 2 weeks. And some of the questions that they've asked me have taken me 10 years to answer. So I work on 2 time frames. I try to give a dirty, quick answer. And then behind the scenes, we try and get the right answer. And it often takes much longer than we'd like. So as Sanjit said, I am a statistician who try to make all my work available. So I have an online book, which I wrote with Wolfgang Huber from EMBL, about our software for analyzing bioinformatics type data. And I want to say I'm a statistician, I'm not a probabilist, and the statistician has a dogma, and the dogma is we start with the data. We don't start with a model. So the data comes first, and that's this X thing, which is punch card from my time. And so you had the data, and then we do various kinds of estimation and create parameters. And from that, we have some kind of probabilistic evaluation of uncertainty. And so that's the statistician's viewpoint, and that's my viewpoint. And you'll see starting with the data is not quite so simple. Usually, when you think of data, you think of features which are given to you that are measured. In this case, actually, the features we have to extract from the data. So we're already in a situation where they're not predefined features. So the subject is microbial ecology. And in particular, I'm interested in HIV and various pathologies associated with preterm birth. And we look at the relationship between the vaginal microbiome and these different transmission of HIV, immunological profiles, and preterm birth. So the difficulties with the data are also that every sample doesn't have a predefined number of reads. So this is the same thing as what happens in ecology. You go out in the field and you decide you're going to listen for birds for 2 hours. You get a certain number of birds, and you go again for 2 hours and you get a different number of birds. So the denominator is not the same. So that's what we call sample depth. That is, you take a sample and you sequence it, and you might have 10,000 reads or 100,000 reads or 1 million reads. And so the denominator is different. So that's very important for the statistics and the uncertainty. And so I'll give you, to be concrete, a study that we did about 10 years ago, which had to do with looking at the bacteria in swabs from 50, we had 49 women, pregnant women, and we set aside 9 as our validation set. And we looked at these 40 women, and we had samples of the microbiome from different sites on the body. And so we had saliva and oral microbiome. And the one I'm going to talk about today is the vaginal microbiome. And we looked at how the bacteria were sort of the co-occurrence patterns of the bacteria. Now, as I said, we have the problem of depth. And so the depth of the different samples means that you have heteroscedasticity because you have different variances, and the quality is very different for these different samples. And this is pretty well modeled with various kinds of mixture models, in particular the gamma Poisson does a good job, and then we can make it a little bit more complicated. And so we were able to do preterm birth prediction using these data. And we can see simple models which do pretty well at understanding what's going on. So we had to be careful, and we did these noise models, but we used also hierarchical mixed models. We used a finite state Markov chain to try and understand the passages in the different states. And we were able to do differential abundance. So we found, in some sense, 2 or 3 species which were really associated to bad outcomes. Preterm birth, inflammation, and so there was definitely a strong signal there. And this is what work that I did with David Relman and his group. And we developed software as a sort of side, which became important later on. And that I did a lot with Ben Callahan. So the questions we asked are, are there sort of types of bacterial communities that were already seen in previous studies, and are they really stable? That is, we're going to take longitudinal samples. We're going to follow women through their pregnancy, and we want to know whether the states are staying the same or whether they change. And can the alterations, the changes that we see, or the types of vaginal communities help us predict preterm birth? And how early does that occur? So could we predict preterm birth at 2 months or 3 months, that was going to be something which was risky? So those are all the questions. So here we had data that came from what we call 16S RNA. So we take one gene, and that particular gene serves as a fingerprint. And that fingerprint tells us pretty much what species of bacteria is present, and we can measure it. So these are the different bacteria that were present in these samples. And then the columns are the samples themselves. And as you see in the columns here, the samples over time, they're repeated. So you have some of these are clumps, it's the same woman, and some of them are separated. And we have some different, what we call community state types, in that, for instance, when you have a lot of lactobacillus, we're in what we call community state type 1. And community state type 2 is the lactobacillus gasseri, and 3 is iners, lactobacillus iners, and 5 is either iners or Jensenii. The one thing that you don't have is a characteristic bacteria for this group 4. And this group 4 is the one that we're going to concentrate on, because the top line tells you whether there was preterm birth or not. And so the pink and the very bright pink are the preterm births. And you see that 4 is associated with most of the preterm births. So we're interested in that. And this is actually a case where diversity is bad. So in many cases in ecology and in microbial ecology, if you have a diversity of microbes, it means that you have stability. It can change around. In this case, diversity is pathological. So we have this group which isn't well characterized by one species, which is the group where there's most of the preterm birth. So that's what we're going to be interested in and try to understand more of. And so we have these types at the top here, 1, 2, 4, 5, and I've colored, I actually split 4 here into 2 groups because they're not homogeneous, but the pink and the red are the group 4. And you can see, so these are the pregnancies, and the length of the pregnancy, it ends with the bracket at the end. So you see the short pregnancies are here. So you see a lot of red in the short pregnancies. And then we can see that there's transition. We made a markup where you can make a Markov chain and you can look at probability of transition back and forth between the different states. That's the beginning of trying to understand the dynamics of this. This is a sort of zero model, but it's quite useful. And so this is the longitudinal data. And you can also make a sort of dual network. That is, you can look at the taxa, which are co-occurring, and between the different samples, the sharing that's happening. And you can see again that it's really in group 4, there's very little which is shared. There's very little clumping. So it's a misnomer to call 4 a community state type because it's just the absence of type. And all the others are types. But that's the one that we're going to be more interested in. But it was useful to make this first model, and it was something that the medical doctors really liked this model. It's something that they could understand pretty well. And so it just says that community state 1 is very stable, so is 2, and 4 is the least stable. But you could get there from any of the other states, not so much from 2. But so this Markov chain is a very simple Markov chain, but that was the first paper that we published about this. We had a certain number of conclusions, and we found these species. In particular, we had elevated Gardnerella, which seemed really important to look at in terms of preterm birth. So we did actually a validation set that we hadn't looked at, the 9 other subjects, and we did separate diagnostics for these 9 women, and 4 of them had developed preterm, and they confirmed the results of our model. So it seems that there's a true effect there of the Gardnerella, and we got interested in it. Now we had problems because I told you with the 16S, you can get fingerprints sort of at the species level, but you can't get anything at the strain level. And as some of you might know, strains matter. That is, different strains of E. coli have different pathologies. And it's the same for Gardnerella. That is, the strain information is important, so we'd like to have more data. And for me, all this information isn't, that has to be integrated or put together in a way which is easy to code with, so we can subsample. And so that had interesting consequences from a software viewpoint, and it motivated what we did. So the next study that we did was a replication study, and we refined the way in which we defined the strains. And so I'll explain to you what we did, but it did show that we had very good replication. That is, we did a multidimensional scaling, the relative positions of the different species, and you see that they're very, very similar. And so this was the original study, the Stanford data, and this was from Alabama. And so we have something which is quite similar. Here we differentiated, we used extra information to differentiate the different strains of Gardnerella. And I'm going to tell you how we were able to do that. We also went in and did what we call metagenomics. And so in metagenomics, it's different than the microbiome data. You take a shotgun and you take every piece of the genome of all the bacteria. You don't just look at one gene. You could look at all of them. But it's an enormous amount of work to recreate the genome of the individual bacteria. And Daniela Goldsman spent 4 years assembling from the data all the different Gardnerella strains. So we looked at the phylogeny of the Gardnerella strains, and we saw there are in fact 3 strains that we're interested in. But that was done not using the 16S data. So what we wanted to do is to avoid doing all the metagenomics and spending 4 years assembling the genomes, but have something with higher resolution. So we realized that we were losing information by doing the fingerprinting in some sense of the microbiome data. So we realized that we have to use probabilistic methods in order to do that, and think very carefully. There was a lot of information discarded in the data in the standard bioinformatics pipeline. And we also created special objects. So I created a software package called phyloseq, which is in Bioconductor, which is used as a standard for storing. You have the phylogenetic tree, you have the table of the contingency table data, and you have all the clinical data, and you want to have it all bound together so you don't lose any of the information. And then if you I program in R, but if you make reproducible workflows, you don't have to care about choices so much. People get very hung up about tuning parameters. And I always say, just choose one. It doesn't matter. We can come back. The script is the same. You're just going to change one line in the code. It's okay. Because sometimes there's a lot of dithering about, oh, well, if I choose something wrong, but you know, it's code, you can just do it again. So the big hurdle that I said was we lost resolution, we lost information, and I'll explain a little bit about why. So the data themselves, they are these reads. So I've made these reads up there, which are these short sequences, 200 and 300 base pair long. And we realized that we could do de novo discovery of the strains from the data without using any kind of reference in a probabilistic way, which was much more efficient and didn't lose any information. So this is work that I did with Ben Callahan. And the idea is the following. Okay, these are the data, and you start by looking at the ones which are all agree. And so say you have this many purple. So I have 8, 9, 10, 11. And the ones that are sort of similar, they might be a little bit noisy versions of these. And I'll explain a little bit more about that. But that's how we do it de novo. We're going to do it probabilistically. So I call them, for the time being, they don't have a name. We just say ASV1, ASV2, ASV. So we call them amplicon strain variants. And one of the things that we noticed was there were a lot of papers about, you know, the black matter in the bacteria, in the sense of that there was millions of new species. Actually, a lot of those species were due to errors. There's a lot of sequencing error that goes on in the lab, and there's also contaminants and chimeras and things like that. So there's a lot of fake species and noise which is occurring. And I think we learn a lot, and now, you know, ChatGPT, it has made everybody realize that language is a very good metaphor. So think about what happens for words, for language, and you want to know how many words a person knows. So you go off and you say, how many words does this person know? And, you know, this person doesn't use a word processor. They write everything down. And you start sampling the words. And it looks as if they know a lot of different words, but in fact, they're just very bad spellers. I live with one. So, so how many real words? Well, that's of course what Google does. That is, when you type something in, it says, do you mean blah, because you made a spelling mistake. And you type something in that practically never occurs. So you want to use the frequency information to infer the spelling and to infer the variant, the strain. So that's what we did. So in this data2 denoiser, we use the information and we have some kind of error model. Now, I want to make a little bit of a contrast to what was being done, the bioinformatics pipeline that was being used as the standard for the microbiome, which seemed to be losing a lot of information. And this was what happened. You have strains and sequences of very unequal numbers. And if you have a noise model, these noise models are the same, but here, say you had 10,000, and here you only have 1,000 of those. So the opportunity for error is much bigger in the blue group. And the standard, what was called operational taxonomic clustering that was used by many of the programs, Time, Mother, and so on, decided that in order to define a taxon or a species, I'm just going to say 97% similarity. Okay, I'll just decide I'm going to have something which is 90, without taking into account how many copies you have. Well, if you have 1 million copies, you have many, many more opportunities for error. So this is not the right model. You don't want to fix a ball of a fixed radius. What we did was we made a model and we denoise. That is, we make a probability model and look at whether it's more likely that this new strain that we haven't seen, is it close to a very common strain? If it is, then it's probably just a mistake, you know. And so you can, when you had the original sequences, then errors creep in. These are the reads that you get, but you can denoise them, and that's how we did it with data2. Or you could make OTUs, and that was the standard bioinformatics pipeline that people were working with. So the error model that we do is you look at a probability of substitutions. Of course, you don't have very much to start with, so it's just like the EM algorithm. You get started with the proportions that you have of all the sequences that are actually equal, and then you start building up the probability that something is a little different, and you update it. You keep until you converge. And you can, in your probability model, you put in all the information you have. So we have quality scores along the sequence, for instance. We have also batch effects or runs. So that solves some of the leakage of information, and now it's a standard. In microbiome work for 16S, most people use data2. And I think it's, so there was the idea, the algorithm is pretty simple, and it was the fact that Ben spent more than a year making a good program. So it's an R package, but under the hood, it's all C++. It's very efficient. That is, you can do very large data sets, doesn't put everything in memory. And so it's the standard for doing millions of reads at a time. Now, this was the output that really was very important to us. That is, if you use the standard workflow, all of this says these are the same strain. This is just one OTU. There's no difference. With the denoise pipeline with data2, we had strains, and so everything that's contiguous here is measured from the same patient. And so you can see that there was a switch here from being red and green to being L2 and the green one, and L3. So you can see that has the strains changed. So this is very useful to us because we can follow how the strains are changing. And we're currently doing studies in South Africa where we're trying out different strains of Lactobacillus crispatus so that we can establish the right communities and bring down the incidence rates of various the different kinds of problems, preterm birth and HIV transmission, both depend on this environment, this vaginal microbiome. So the strain information has been very, very useful to us here. Now, the output, and as a statistician, I want to stress this, is we have different samples, and the sums of the columns are all unequal because you have different numbers of reads. And these weren't given to start with. That's something that I developed, that we developed from the data. Okay, so you've created features, new features from the data. And in general, the number of ASVs is way, way bigger than the number of samples. So that's a very high dimensional problem. And so the model has to be non-parametric because of the large number of features. So it's important to remember that this is a contingency table. Contingency tables, you have counts, and the data themselves are these counts. That's what the data are. So if you're saying that the data are the proportions, you're lying about the data. So you can't lie. You can't just change and say, oh, the data are compositional. No, the data are not compositional. The data are a contingency table, and that's what the data are. You can say, I'm interested in estimating the proportions, but that's a parameter that you're doing down the road. The data themselves are counts, and you need the counts because sometimes you need to know, you need to remove contaminants, and it's much better to have the counts. You need to estimate the depth bias for different samples. So you always have to keep the column sums. And people think I'm crazy to be saying this again and again, keep the column sums. But the number of times that I have biologists come and see me and they say, I have this data and they're all proportions. And I ask them, where did the proportions come from? And they all have denominators, but the denominators got lost. And you can't even find them again, so you don't know what the variances is in the binomials. So, you can make transformations which make the sample depth, make a transformation by ranks, or you can do Anscombe, or you can estimate size factors, so the depth isn't a problem. But the raw data are the counts. Okay, so it's important because you need all of this information. This leakage makes it that you can't quantify uncertainty as you go along. So then what we do once we've done data two is we combine all of this in this complex data structure with an S4 object. And this phyloseq enables you to manipulate that very easily. This was work with Joey McMurdie, and he was actually a soil scientist when I met him, but he wanted to do dry lab stuff, and he became a statistician. And he made the implementation, which makes it so that a biologist can just type a few lines and have access to ggplot and make nice networks. And we still have a community of people using this software. Although I never got any support for this software because I don't look like a computer scientist. So. But okay, so the next question that comes up is, how do we model the different species? So you could say, oh, it's a contingency table, and the ASVs, it could be categories, and I could just do a multinomial. But unfortunately, in the multinomial, you can't build in the type of multivariate dependencies that you have. So when we were doing the pregnancy, we saw that the lactobacillus, if you had it, you practically had none of the other species. So the lactobacillus and the Gardnerella are completely exclusive. So it's negatively correlated. So you can't use an ordinary multinomial model. In many of the species, you have co-occurrence because one species needs what the other species is creating. We call that syntrophy. And then we have also, you know, some different types of small communities which all are producing things for each other. And so you have like a clique structure. So the ordinary multinomial model don't work. And you can benefit from thinking of it a little bit like words because some of the species are kind of interchangeable. And so some of the strains, some replace others in the same way as in a sentence, you might have synonyms. So again, I think it's the metaphor of language is pretty useful in understanding. And so we decided to use the mixed membership models, the topic models that we use for language models, because in fact, in clustering, you could only have one cluster for each sample, say, one type. And it stops you from seeing any transition. That is, it's like a 0-1 law. So if you had, 51% of one type and 49, it's about to change, but you're not going to see that. So it's better to have a probabilistic mixed membership model, and topic models are ideal for that, because you can see the bacteria sort of self-organize in guilds. And so this is the parallel that we built with Kris Sankaran, in which for us, we have biological samples, and then you have a document, and the species are replaced, the species is what you have in terms of the bacteria, and you get them from the reads. And the term, in some sense, nowadays we'd say the token, comes from the words. And then the equivalent of a topic is a community. And then you have a corpus, and that's an environment. So it's a useful model, it's a useful statistical model. It was first done actually for genetics in mixture models. John Pritchard and Matthew Stevens and Peter Donnelly, they did it in 2000 for having genetic mixtures. And then it was done, this latent Dirichlet allocation was done by David Bly for language. And so you can do the posterior inference with a very sort of standard implementation of collapsed Gibbs sampling. What it gives at the outcome is that you have observed microbiome mixtures of underlying community types. And so instead of just having a cluster with a hard boundary here, you have each point is a mixture of the different topics. So the ones that are here, they have a very high proportion of topic 2 and very small of those, but you have more information. And you're keeping some of the uncertainty with you all the time, and it's much richer type of model. So of course, the statistical model is, I like to think of it as being dual because it's both a mixture for the samples and a mixture for the bacteria. And so for the bacteria, organize in community types, and then the samples have a mixed membership of these different community types. That's the sort of standard way we'd see it. Now, the output that we get here, this is an example of perturbation after taking antibiotics. The topics that came out and the proportions here, what you see is a big drop in topic 1 after about 3 days. So this is the first course of antibiotics the patients had, and the second course of antibiotics was here. And you can see the behavior of the different topics is different after the first course as after the second course of antibiotics. And so we have these, you know, these are the top 4 topics. And you can look at, you know, which are the species which are most prevalent or explain the topics the best. And so we have these different types of families of species in the different topics. So we have an interpretation which is useful to the biologists. And here we just have some typical abundance profiles across the different topics. And as I said, you know, topic 1 is the one that really drops after taking the antibiotics. And if you look at the individual species, you see it pretty well. Now, one of the questions that came up a lot when we were doing these topic analysis was, how do you choose the number of topics usefully? And there's a very nice paper that was written by 3 of my graduates. Well, Kris and Julia were my graduate students, Laura was my postdoc. And they wrote a package where the inspiration is to think, when we're doing clustering, it's very useful to look at hierarchical clustering because you can sort of decide where to cut off. And you can do the same thing with topics. Here we have the different models, and we have the total mass and how they are sort of shared between the different species here. And you can see how they disassemble and reassemble. And so, you know, this is 7 topics, this is 8. And you can see that, you know, this topic, for instance, this stays practically constant or, you know, composed of the same species right through the process. And so here, and so this is a method for aligning in a network all the different topics so that you can understand what's going on, how much they split and come back together, and how many topics you think, you know, you could have. So in this case, we chose 11, and it seemed to give a good representation in some sense of topics which were pretty stable across several numbers of topics. So that's in a package called Alto, which is, and we use this very much in comparing. So if we look at, we have a study in which we're looking at pregnant and non-pregnant subjects in the same way as I was explaining before, but instead of doing community state types, we're doing topics. And that's especially important for this non-lactobacillus group. And here you see that we have this group, which is sort of the bad group, the one that we always look out for, that helps diagnose problems. And the Gardnerella, Apobactobium, Aerococcus, they're all together, and they've been together for several K and 9, 10. But they also occur, these 2 occur, this is a different species, but the Gardnerella with Apobactobium also occurs in the pregnant subject. So we're able to do comparisons between groups, but at the topic level. And in some sense, you can make the variables, that's why I say you have hidden or latent variables, you're making the variables which are easier to interpret yourself. So we have that choice. And we have also this topic modeling helps. In a paper we wrote with Pratheepa, it solved the problem what we called strain switching, which was when you do a test to see if these 2 groups are different, if one strain is switching back and forth, then you won't see those strains as significant. In fact, they're just like synonyms. And the strain switching problem goes away if instead of you working at the level of the individual bacterial species, you work at the level of the topics. So, of course, our goal is better reproducibility, and we do everything with R. We have R marked down. And I have, you know, quite a few supplements where all of this is written out, and we try to keep all of the packages that we make up to date, which is a lot of work because the packages they depend on often change a lot. So we have reproducible research supplements. In some of the packages that I talked about, phyloseq and Data2 are in Bioconductor, and then the topic one is on GitHub, and the decontamination one is very popular. And that's something which also uses the simple mixture model and enables you to find whether you have contaminants in your species. And so the book is online, and it's also a lot of work to keep it up to date. So I'm happy when people use it. There are more complicated aspects of the data, which I won't go into too much. I'll take some questions. But one of the things that we have in the Gates study and many of the studies is that we don't only have the phylogenetic tree and, say, longitudinal data and samples taken over time. That's just what I was talking about, maybe the pregnant and the non-pregnant. That would be just the top part of this. What we also have is we often measure gene expression, and nowadays we do RNA sequencing, and we do metabolomics. So we measure proteins, and we have the networks of proteins, and we also have them over time, and then we have the samples. So there are 1,000 names for this. The computer scientists called it multi-view data. The way I came into it, the subject with multiple tables. So I was trained in the French school, and the French are interested in food and wine, as you know. And so some of the data that we had was you have wines, those are your samples, and then you have, say, chemical measurements on the wines. And then you have the wines, and you have the geographic area they came from. And then you have the wines and the composition of the soils where the wines came from. And then you have the wines, and you have judges making judgments about the wines. So you have multiple tables. So that I think that one of the first areas where they did what we call multi-way data was in food science, and that was in the 70s and 80s. But we're still doing this. Now we have multi-omics, so you have all the different omics, but I don't like the word multi-omics because in some of the data I analyzed, some of the data are images, so it's not an omic really. You know, you have images and spatial data, but you have this idea that you're going to have heterogeneous data and you want to put them all together. You want to see whether they have a relationship or not. So that's an important thing. So here, this is an example of the multi-domain data assembly and analysis. And it's just to illustrate what I was saying about reproducibility. So this is a study that was led by Laura, and Pratheepa participated in. And we were looking at topics and of the vaginal ecosystem in pregnant and non-pregnant women. And so we wrote this paper, which has now appeared at the Royal Society, Proceedings of the Royal Society. And this is an example of what happens today. That is that you have very complex data and you just have so many choices. So if I computed the combinatorics of the choices that we made in the ways of computing the different distances between the data and doing all the different possible cutting and slicing of the data, there are more than 200 million ways of analyzing that data. And post-selective inference and multiple hypothesis testing will do nothing for you. You know, it's not going to solve the problem of branching paths. You have too many ways, and the only way of solving that is just showing your work, and somebody can rerun your code and just make another choice for you. So that's what we did. And so the supplementary material was the code plus the PDF generated by the R Markdown. And that's why I say my postdocs and students are very patient people, because this is a supplement which was 227 pages long. It's just too long. But there's everything there. That is, anybody could say, okay, you chose Bray Curtis distance, but I want to choose Jacquard distance. And so what will it change downstream? And you can just do it because you just one line of code to change. So that's the motivation in some sense for saying, I don't think we can solve the multiple, 200 million ways because that, you could say anything. When I started at Stanford, there's a consulting workshop, and it would be throngs of biologists who come and see us, and they would say, my p-value isn't significant. What can you do for me? And so we were clever. We knew you can, there's a whole array of transformations you can make on the data, and it helps with the p-values. And so we were very popular. I can make any p-value significant. If you let me drop out 5 points and make the transformation, I'll make your p-value. It doesn't mean anything. The actual findings are not reproducible. So I think the only way is computational, that is, show your code. And so we have all kinds of structures now, and this is a very useful one. We have the S4 classes for putting in all the data and all the information about the samples. And this is some of the output from that. That is, in this case, we had the metabolomics, we have different proteins, we have the different pH, we have the species communities, the lactobacillus dominated ones are in blue, and the non-lacto are in red. So you can see the pH is much, much higher in the non-lacto one. And so you have all of this information that you could integrate in a useful way. And I'll talk a little bit more about how you have to do the registration for that tomorrow. But I think that one of the things that the bioinformaticians don't understand about is data are not uniformly distributed. And so you can't just drop information without it having an effect. So there's distribution. There's the problem of lost information and the problem of black boxes. That is, you need to know exactly what was done to your raw data. Now, I have a hero who is a friend of mine. That's Don Knuth. He's the Yoda of Silicon Valley, or that's what New York Times says. And he says, premature optimization is the root of all evil encoding. But I have my own. And what I find is that people are trying to summarize the data. Premature summarization is the root of all evil in statistics. And for instance, in the case of clustering, it doesn't cost a lot to add that you had 49% of one type and 51% of another. And that's what you need to do. That is, you mustn't summarize and say, oh, it was cluster 1. It's not 0, 1. And so I think that we need to, keep all the probabilities, keep all the information, so that we can then put in the uncertainty. And so what I learned is you have to understand the raw data. And I don't know, this generation probably didn't watch Dr. House. But Dr. House, it's exactly what I find with the biologists. They lie about their data. They tell me, this is the data. No, that's not what the data was. What was the raw data? You've just cleaned it up. And often in the cleaning up, oh, it's relative. No, it's not. You lose all the information. So you have to come to terms with the messiness. And so the other thing is, of course, the one size doesn't fit all. We do studies on the microbiome where we're interested in rare species. You have to go to very, very deep. You have to have sample sizes of 10 million reads if you want to look for a very rare species. If you're looking for the core of the microbiome, you probably can get away with 20,000 reads. So design is very important on the question asked. You're not allowed to ask all questions. You have to say ahead of time which are the questions you're interested in. Choices are not forever. And if you have enough of the information that you can reconstruct everything and the code, you could make other choices. So it's important to have your code. I like hierarchical models, and I'm going to talk tomorrow about more the registration and alignment. And an analysis is only as good as the explanation in the software. So one of the problems in statistics is there's a lot of publication of theoretical results, but it's unusable by the biologists. So you have to make something, that's why the topics has been so good for us, because we could explain it in terms of language, something that they can understand pretty well. And so finding metaphors and explaining it to biologists, then they'll adopt it. But if it's obscure and you just say, oh, it's math, trust me, they're not interested. Because why? Because they have to stand up at the next immunology conference and explain it. And that, you know, trust me, doesn't work there. So telling a story is much better. So I, you know, thank my lab group, or this was a picture of my lab group before COVID, and David Relman, with whom I do most of my work on the microbiome. And thank you for having me.